Hello, everyone. This is Kelly Clausen, and on behalf of Q1 Productions, I'd like to welcome you to today's webinar and thank you for joining us today. The webinar will discuss IVDR Survival Guide, common pitfalls encountered in complying with the regulations requirements. We are very lucky to have Smriti Harihan as our speaker today. Now, without further ado, I would like to briefly introduce Celegence and our presenter. As mentioned, this is for IVDR Survival Guide, Common Pitfalls in Complying with Requirements of IVDR. Here's a brief introduction on Celegence. Celegence has headquarters located in Chicago, but they have other locations in the US, UK, India, Netherlands, and Romania. They are ISO 9001 and 27001 certified. Their mission is to provide regulatory value-added services for life sciences company that improve compliance, increase efficiency and productivity, and reduce costs. Here's a couple of their clients as well on the slide. Their global approach, this shows their Celegence offices and partner offices across the globe, as you can see, located in US, UK, Brazil, India, China, and Japan. Here is an overview of the medical device and IVD services available from Celegence, including their capabilities, medical guidelines coverage, therapeutic areas covered, their experience of their teams, and some of the achievements on behalf of their customers, including notified body documents approvals for over 300 unique products or families. If you would like any further information on any of these services, please reach out to the Celegence team by using the email provided at the end of this presentation. And for our presentation today, we will be looking at the IVDR Survival Guide, a timeline for IVDR, types of clinical evidence, routes for conformance for SVR, CPR, APR, and systematic literature research searches. A little bit about our presenter. She has over 10 years of medical writing experience, including medical devices and in vitro diagnostics. She has experience spanning different therapeutical areas and the different risk classes of our devices. Now, without further ado, I will pull up our presentation.
Hi, everyone. Welcome to today's session on IVDR Survival Guide. I'm just going to go over a couple of tips and tricks on what we can do and how we can write various PMS documentation for in vitro diagnostics to make them compliant with the recent IVDR regulations. I'd like to start off with the timeline for the IVDR. As we all know, recently there was a postponement of the timeline. So the regulation was first released on 26th May 2017, after which in 2022, all IVDs that were placed on the market and Class A non-sterile devices had to be made IVDR compliant. And 25th, now the current timeline says that from 25th May 2025 onwards, all IVDD CE certified devices must comply with IVDR. So the existing certificates will not be valid anymore from that date onwards. The Class D self-declared IVDD devices also must comply with IVDR. And then from 26th May 2026 onwards, the Class C self-declared devices will have to comply with IVDR. And finally, from 26th May 2027 onwards, all Class B and A sterile self-declared IVDD devices must comply with IVDR. So this data has been taken directly from BSI's website, and these are the timelines that are currently in place. All the IVDs on the market that are currently that did not require certification under IVDD must be checked again to see if the classification has been changed, and we must reevaluate if under the current IVDR guidelines they do need to be certified by a notified body. The regulations have said that the sell-off period for self-certified IVDs that are already on the market is removed. These devices, they can be made available on the market without any legal time restrictions. And for all in-house devices, the requirement to justify that an equivalent device is not available in the market is postponed until May 2028. So some of the important definitions or key points or checkpoints that we should keep in mind while drafting PMS related documents for IVDR are intended purpose, clinical benefit, clinical evidence, and then performance evaluation. So one of the most important PMS documents that we draft for an IVDR is the performance evaluation plan and the performance evaluation report. These encompass all of the points that need to be satisfied for an in vitro diagnostic device to comply with the GSPRs under the IVDR. So, in order to have a complete performance evaluation, it's important to understand what what is the intended purpose of the device, what is the clinical benefit, and what exactly is meant by clinical evidence under IVDR, because the requirements for clinical evidence under IVDR are different from what is required under the MDR. So for the intended purpose, the definition is quite simple. It's the use for which a device is intended according to the data supplied by the manufacturer. And this data is either on the label or the instructions for use or any promotional materials that accompanies the device. This intended purpose is the basis for all of your performance evaluation. Your entire performance evaluation will be focused on showing that your device achieves its intended purpose. And by achieving its intended purpose, it has a clinical benefit. The clinical benefit here is a positive impact that the device has on the patient by nature of its intended purpose or its function. The clinical evidence here refers to any clinical data that you are using to demonstrate that the device achieves its intended purpose. And it is essential that this clinical data is of sufficient quality and amount to show that the device is safe when it's used as intended and it does have a clinical benefit associated with its use. So, for a performance evaluation, there are mainly three key points. One is the scientific validity, one is the analytical performance, and the last is the clinical performance. So when we talk about clinical evidence, we talk about providing clinical evidence for all these three components. However, clinical evidence majorly supports the analytical performance and the scientific validity of an IVD, versus the clinical performance is mainly supported by clinical utility. In some scenarios, it is possible to provide clinical evidence to support the clinical performance of an IVD as well. We'll just go over what we mean by the analytical performance, scientific validity, and clinical performance of an IVD. So, analytical performance, as the name indicates, is just the ability of a device to correctly detect or measure a particular analyte. Since this is not related to a patient or it's not related to a disease as such, we're talking about an analyte and how our device correctly measures it. Most of the data to support the analytical performance will come from in house testing, will come from lab tests. It doesn't come from outside sources like published literature or clinical studies that are being conducted on the IVDs. So most of the analytical performance is in-house testing. Scientific validity is the proof of an association between the analyte as well as the clinical condition. So the data to support this comes from many different sources. We'll be covering that in the slides going forward. Clinical performance is the ability of a device to show that there is a correlation between a particular clinical condition or a physiological or pathological process in a particular target population and an intended user. So, when we talk about a relation between an analyte and a clinical condition, it's a scientific validity. Whereas when we talk about the device with the clinical condition, it is a clinical performance. So that is a very small and minor difference between scientific validity and clinical performance. But the kind of data that you would use to support the scientific validity and clinical performance changes very greatly because of this small difference. So the clinical evidence, you will be using it to support the intended purpose of the device, as mentioned earlier, and that will form the basis for your performance evaluation. So the very first step is to clearly define all different aspects of your intended purpose. There are several different things that you must consider while coming up with the intended purpose of your device, or while creating a statement for your intended purpose. Some of the major things that you would have to consider are: what is the analyte, what is the function of your IVD, which clinical condition or physiological condition or pathological condition within the body does it associate is it associated with, is the device automated or not, are the results going to be qualitative or quantitative in nature, what type of specimen you would be using to conduct your testing, what is the testing population, and who is the intended user. So, on the basis of these different requirements, you will be able to frame a concise and well worded intended purpose, which you can use to draft your entire performance evaluation. If you have all of these key requirements in place, you will be able to determine what type of data you need, from what is the best source to collect this data, um, what data do you already have to support each of these different requirements, and what additional data you require. So it's very important to identify each of these requirements well in advance of your performing your performance evaluation. So I just go over what each of these means. So analyte, as I mentioned earlier, was what is your device detecting. Function is what how does it detect the analyte, what does your device perform. So your function is mainly is supported by the scientific validity, as well as in part by your clinical performance report. So one of the other important factors that we consider is the clinical condition, or the pathological condition, or the physiological condition within the body for which the device is being used. The reason being, this will form the basis of your state of the art. And state of the art is one of the very important sections that will also have to be covered in your performance evaluation report. Unless you have a very strong foundation on what the disease condition is, how it progresses, details on what other devices are available to perform the same intended purpose, how else can you diagnose the same condition, cannot be determined. So it's very important to lay down exactly what pathological or physiological condition your device is being used to detect or measure. Next is to understand if your device is automated or not. When a device is automated, it is important that there are specific tests in place to demonstrate compliance with the GSPRs. For example, you will have verification validation testing, or algorithm details that need to be presented in the performance evaluation report for an IVD that is automated. That obviously will not form a part of the performance evaluation for a device which is not automated. Um, the next step is to determine if your device is going to provide qualitative or quantitative results. So if your device just is going to give you whether a positive or a negative, versus it's going to give you a range or a particular value, the testing requirements will be different for both of these conditions. Uh, the accuracy, specificity, diagnostic sensitivity, these are things that you will have to consider and determine what is applicable to your device based on the type of output that it is giving. Similarly, specimen type. A lot of the times we refer publicly available information from published literature. So it's important to note down what specimen type they've used. If it is not in accordance with what you've identified in your intended purpose, then it will be considered as an off-label use, and the data cannot be leveraged for the performance evaluation report. Um, Similarly, you'll also have to consider the testing population and the intended user, the requirements for a lay person using a device versus a trained clinical professional using the device are very different. Even the risks involved are different. So the way the data is presented within the performance evaluation will depend on the testing population and the intended user as well. So here, clinical evidence, like I said, means clinical data and performance evaluation results that are pertaining to a specific device. And the article within the IVDR very clearly states that it must be sufficient amount and quality to be able to assess if the device is safe and it achieves its intended purpose and clinical benefits. And then Article 56, Section 2 says the clinical evidence should support the intended purpose of the device as stated by the manufacturer, and it should be a continuous process of performance evaluation following a performance evaluation plan. Um, so, like I mentioned earlier, the three main components of a performance evaluation are the clinical performance report, the scientific validity report, and your analytical performance report. Analytical performance very largely comes from in-house testing and in-house data and does not require a lot of data coming from outside. So I won't be focusing on that in this particular presentation. I will be focusing only on two main things, which is the clinical performance and the scientific validity. So, clinical performance, as defined by the IVDR, is the ability of a device to yield results that correlate to a particular clinical condition, a physiological or a pathological process, and are in accordance with the target population and intended user. So there are several different routes available to generate or collect evidence for support of your clinical performance report. The three most common routes are either conducting clinical performance studies, collecting evidence from peer-reviewed literature, or publish routine diagnostic testing. The most commonly employed route is through peer-reviewed literature. Reason being, it's easily accessible and it's one of the easiest ways to collect data. Clinical performance studies are usually very hard and cumbersome to perform, and most manufacturers tend not to go down this route unless their device is significantly high risk and there is little to no data available either through peer reviewed literature or published routine diagnostic testing. So for new IVDs that are based on new technology that hasn't been on the market for long, the only route available or the safest route available would go through clinical performance studies. For any studies that are performed on IVDs, they would have to be compliant with the ISO 20916 standard. Only if they are compliant with this will they be able to be added to the performance evaluation report and considered as clinical performance studies, and the data can be used and analyzed. If they are not compliant with this standard, then they will not be considered as clinical performance studies. So, apart from just conducting the study, before even starting off a study, it is important to get a couple of points out of the way. One is to assess what type of device is it. Is it a near patient device? It is a self-testing device. Do you need a separate validation to be conducted? What type of study needs to be there in place to show or to demonstrate clinical evidence? There also has to be a risk analysis, and the intended purpose should be clearly identified by the manufacturer, and the environment in which a study to be performed also has to be identified. But again, this is like the last option for most manufacturers. It's only used in case the device is extremely high risk and there's no other viable data available. The most common route, as I mentioned earlier, is through peer-reviewed literature. So it is important to have a systematic search done in place where a research question has been clearly identified, the objective of the search has been clearly identified, and the search has been executed for a suitable duration of time on scientific databases. And if the search is reproducible and it's concise and simple and it's easy to follow and it is yielding a good number of relevant results, then it's considered a good systematic search strategy, and it can be used to collect suitable peer reviewed literature to support your CPR. In this scenario, uh, It's usually possible where devices have been on the market for a considerable amount of time, or they are based on a technology that has been on the market for a considerable amount of time. And one of the points to consider is that it's not always necessary to search for publications specific to your device or your brand or your specific manufacturer. Since IVDs they're very similar to each other, and most of them are based on the same technology. We have seen instances where we were able to leverage data from similar devices without claiming equivalence. Uh, Simply because the devices are so similar to each other, and we were able to demonstrate and prove that there is not much difference, nothing that would affect the safety or performance claims of the device. And the last route available to establish or to show evidence in your CPR is through published routine diagnostic testing. So there are often cases where routine diagnostic testing has been conducted and then retrospectively analyzed, and there are publications. These are not peer-reviewed publications, but it's just an experience of a particular testing center or a particular clinician or a particular trained clinical professional with specific types of devices in certain conditions. And these publications are also acceptable and can be used as evidence within your CPR. So scientific peer reviewed literature, you can consider journals. You can also consider posters from conferences. This won't be considered high quality data, the posters from conferences, but you can still consider them as one of the sources of data and then use it to show that there is some uh, data related to what you are talking about. So, data that shows that your device is safe and performing as intended, but it won't be considered as high quality data. There are several different databases that you can consider. There are a few that are specific to IVDs, but they're not very good quality. So using generic databases like PubMed, Embase, Medline, and Google Scholar is good to collect data for your clinical performance report. There are also several websites where there are guidance or documents from official websites that are available, where you can look for standardized testing results, or where you can look for guidances on ranges or accuracy or cutoff values that are to be used for establishing that your device is accurate and performs in line with industry standards. So some of these official websites are MedTech Europe, IMDRF, WHO. You can also refer to textbooks which are good sources to demonstrate what is the industry standard, or what is the current acceptable limits or cutoff ranges or values for certain devices. um, the other source is published experience through routine diagnostic testing, as mentioned. So usually in these scenarios, any issues that are identified when using a device, um, uh, it can come through multiple sources. It's either information that has been issued for public safety, information that is submitted online that's not yet been accepted. It's information that's been put out by a hospital or a laboratory. It may either be formally done, or it's not officially done, but it's just put out onto social media. Even that can be considered as sources of information and can be considered for your clinical performance report. And lastly, PMPF data, that is post-market performance follow-up, also for other devices, if available online, if you can get your hands on that, then that also can be used as a part of this data input. Next, I'd like to go over scientific validity. So, similar to your clinical performance report, you have a scientific validity report where you have to demonstrate the association of an analyte with a clinical condition or a physiological state. And similar to the CPR, the SVR also has a couple of different modes through which you can establish conformance, or through which you can establish or provide evidence for your SVR. You Here also, there you can consider either published literature, or you can go with textbooks or guidelines. If the device is very well established, if the clinical condition and the analyte that of interest is well established, if it's well known, then there's no need to do your complete SVR. You can just cite or point towards these guidances or um, previous publications that show that there is a link between the analyte and the physiological condition. Performing the entire systematic search and summarizing and appraising the data is not required if it is a well-known condition. For example, there's no need to conduct an SVR for, say, a device where you're testing blood hemoglobin to check for diabetes. The link between blood glucose, hemoglobin, and diabetes is well known. So establishing that this connection is not required. Some of the things that you would have to consider while doing an SVR is the analyte claims, the condition claims, and the testing population claims. So before setting off on an SVR, you would have to determine all of the analytes that are involved. So if your device is, if your clinical condition has multiple different analytes that can be used to detect a particular clinical condition, then all of those analytes must be listed, especially if your device is analyzing more than one of these analytes to determine the clinical condition. Next, what you have to consider is the condition claims. So all of the conditions, if one analyte is linked to more than one condition, if it maps to more than one physiological condition, if it maps to more than one disease, then all of these conditions must be cited out and specified. You have to determine at what range or cutoffs these analytes are indicative of this particular clinical condition. So even the range, the cutoff value has to be established in your scientific validity report. It's not just that if this analyte is there, then the disease is also there. If there is a specific value at which occurrence the analyte signifies the presence of the disease, then that value also must be supported through evidence in your SVR. You should, for very broad or generic conditions, like I mentioned earlier, it's not required to conduct a full-fledged systematic search and justify all of the ranges, cutoff values, or even the link between the analyte and the condition. But you have to provide a justification for why this is not required, and a justification for why you are claiming that there is a link between the analyte and the clinical condition. You can always reference international guidances when you're describing a clinical condition of interest. And last thing you have to consider is the testing population claims. So if there is any limitation on the testing population, if there's a specific population where there wouldn't be a link between the analyte, or if there is a specific age that has to be considered, or an ethnicity that has to be taken into consideration, then that has to be called out as well. And literature to this specific effect of why certain populations are excluded, or why specific deviations exist in populations, must be provided in the SVR. So for demonstrating scientific validity for CE marking, or as a part of your performance evaluation report, there are different routes, as I mentioned. One is to just identify scientific literature through a systematic search, similar to what was performed for your clinical performance report. But your search strings will vary because the objective, the research question that you're looking for an SVR is different. Then you will have to appraise all of the relevant data. You will check for suitability and then summarize the articles as required within your SVR. If there isn't sufficient data available through published literature, through textbooks, through guidance documents, or through other linked data, then the option arises to take data from your clinical performance studies. So any studies that are conducted for your device to show it's to demonstrate its clinical performance can be leveraged in your SVR as well, to show that there is a link between the analyte and the clinical condition of interest. If there are still gaps, then it is required that new or additional data should be generated. And Again, this is done through your performance evaluation studies. which are usually the clinical performance studies, and they have to be according to the ISO standard as mentioned earlier. You can also consider consensus or expert opinions. If it's a very new device, first in market, new technology, new clinical condition of interest, then in that case the proof of concept studies can be used for demonstrating scientific validity. Okay. And lastly, as I mentioned, there one of the most important parts of the performance evaluation report is the state of the art. So, apart from the SVR and the CPR, we also have to conduct systematic searches for coming up or developing your state of the art. Your state of the art is basically what is currently accepted as good practice. It is not what is the most forward in technology at this particular time, but it is what is generally accepted as good practice is considered state of the art. Um, so all of the treatment options that are available, all of the diagnostic options that are available for a particular clinical condition of interest will be the focus of your state of the art. So if your device is um, say oncological testing product, then what you will be focusing on is all of the options that are available for cancer detection. For if you're looking at a specific type of cancer, then for that specific type of cancer, all of the available diagnostic options, even if they are not at the same level, you will be presenting a brief write up of all of these options, how each of these options differ from each other, what are the advantages, benefits, and risks against each of these options. How does your device compare to all of these options in terms of benefits and risks? Um, what are the conditions that are used when you're detecting a particular disease? So these different diagnostic options, are they testing different analytes, are they testing the same analyte? If they're testing the same analyte, how does your device measure up in terms of accuracy and other performance standards? So all of this will be described in the state of the art, and all of this data must come from a systematic search that is executed. Again, you can consider multiple scientific databases such as PubMed, Embase, and Medline. It is important to have a very clear research question in mind while performing the systematic search, so that we don't get a number of irrelevant results. You have to keep the hits down to not more than 500 to 800, and keep it as relevant as possible so that we don't spend too much time on drafting just the state of the art. Um, Also, analyzing similar devices or other brands that have products that have the same intended purpose and may be working on a similar technology will also be focused on in the state of the art. And then analyzing any of the risks reported for these devices will help us identify any gaps in our risk management procedure as well. Um, they will help to create a more robust risk control methods and risk management methods, and make our device stronger in the market. So that is the purpose of having a very strong and robust state of the art. So for demonstrating state of the art, again, one of the important things is you will have to consider similar devices. You will have to consider best practices for that particular group of devices that are either same or similar. You will have to consider results of accepted scientific research, or if it's a well established technology, then if there are publications dating far back as well, also can be taken as background for establishing your state of the art. You will also have to consider publications from authorities or working groups from different organizations that deal with that particular clinical condition of interest and give out guidances on how to diagnose a particular clinical condition or a pathological condition. And lastly, you'll have to compare the benefits and risks of each of these available alternate technologies as well as similar devices against yours. um, for this, the sources can be either peer reviewed published literature, textbooks, clinical guidelines, and even recommendations. Unlike your data for your clinical performance, it's not as strict here in the state of the art. You can reference non-peer reviewed articles as well. If there's very limited data, then we can go for non-peer reviewed literature. Ideally, we would stick to peer reviewed literature, guidelines, and textbooks. If you can see, of all three sections, so the SVR, CPR, and the state of the art, the most important factor was conducting a systematic literature search. I found that this process has been uh, made very easy with the use of one of our systems called Captus, which is an offering of Celegence. This system, it allows us to perform multiple literature searches on the software, and we can store it. And then the screening, sorting process, the identification of duplicates, the appraising process becomes a lot simpler. The system has multiple features that make it easy to go through hundreds and sometimes even thousands of articles and then not get muddled up. We've seen a considerable improvement in um, our time spent, as well as the manual errors that occur or while using an Excel sheet or while doing the process manually. They've been reduced when we use this Captus system. There are it it also facilitates collaboration, so multiple different people can work on a single project at the same time, which helps to again bring down the time, as well as act as a mode of connect between teams that are functioning on different time zones, different locations. It's very easy to collaborate and work and then streamline the process when we're using Captus. So in a recent study, it was found that there was nearly 30% reduction in time on systematic reviews when we compared manual to the Captus system. Apart from just the time factor, there are several other benefits associated with the use of Captus. It's integrated with certain databases like PubMed that allows you to bulk import search data. There's also free full text, everything that's available on the link online. If there are full text available for a particular article, it's pulled directly into the system. So manually having to download full text and then sort through them is not required. There are audit trails available throughout the entire Captus system. So any activity that was done, any change in a particular article's status is recorded by the system, and these audit trails also serve to validate your literature search. Oftentimes it's seen that when the search is executed on different days, the number of hits changes. So when it's stored in a system like this and you have an audit trail, it serves as evidence to show that on that date this was the number of hits that was obtained. A lot of notified bodies nowadays are asking for how we validate our literature search protocols. So this particular system helps us in answering these questions as well. The process for reducing bias, the process for deduplication is significantly easier to track with Captus. So duplicates are automatically caught by the system. There's a three-level review that's involved in our team when using Captus, and each person that accesses the system that goes through a particular article, their data is logged. So the audit trails can serve as evidence to show that there were three people who worked on the screening appraisal process, and therefore the bias is reduced. All data has been included without any bias. So validation of the literature search protocols and demonstrating that these literature search protocols are valid and the systematic search is good, and all possible um, modes and methods of search have been conducted is documented. And it's also the number of errors that are encountered while manually typing out filters that were used in a search, or date ranges that were considered is avoided. There's also data collection forms which can be customized to suit our needs. There's also the option to add in as many or as few tags as you want to an article. You can tag an article if your device is screening multiple analytes and you need evidence for each of these different analytes, then you can create a tag for each analyte. And then once the screening is done, all you have to do is filter each of these tags, and then you can see how many different analytes have data supporting them. If at that time you notice that one particular analyte does not have any articles tagged to it, then you know that there's insufficient data to support the use of our device for this particular analyte, and you can go back and perform a focus search. So this is very helpful to avoid last minute rework or last minute scurrying to find evidence. Your information is at hand right after the screening process. It's all documented in the system, and that way you can decide what your next course of action should be. Do you want to go ahead and perform more clinical studies? Do you have to look for more focus data? Do you have to look for more guidelines? It's very easy to make a decision at that level rather than complete the entire performance evaluation process and then have a notice that there's not enough evidence for a particular uh, reason and then conduct the search again. So, for anyone who's interested in finding out more about the CAPTA system and how it works and what kind of benefits it offers, our team is always available to provide demos. So, the details of whom you can reach out to in case you want more information or in case you'd like a demo is available in this slide. So, I'd like to end with this, and if anybody has any questions, we can go ahead and talk about them right now. Thank you.
Thank you so much. I see that we have a couple questions that have come in. We're going to get to those. All right. For CPR requirements, an experience is that clinical performance studies are not accepted as primary evidence. They are considered only secondary to peer-reviewed literature, as any clinical studies of legacy products would not be done under IVDR. Could you please comment on this? Would clinical studies happening now be considered under IVDR?
We've actually had the opposite experience. We've most often been told that the clinical performance studies would be considered the tier 1 level of evidence versus data that you're getting from peer-reviewed literature or clinical studies of legacy products would be tier 2 or tier 3 level of evidence. We find that performance studies, if done in compliance with the ISO 20916 standard, is accepted by the notified body. And if the study is designed specifically around proving the clinical utility of your in vitro diagnostic, then they would definitely accept it as evidence for your device. But you would have to show that it's complying with the ISO standard. If it's not done on human subjects and it's not complying to the standard, then they won't consider it. They won't consider it tier 1 evidence. It'll probably be considered as routine diagnostic testing data.
Please describe the risk analysis required in the clinical performance study protocol. What is the difference between this risk analysis and what is in the benefit risk analysis required for IVDR and ISO 14971?
So for in vitro diagnostics, since the main objective of your device is to garner data that will be utilized by the clinical professional or whoever is getting that data to further implement or design a management or a treatment strategy, the amount of risk associated with an IVD is significantly lower than a medical device. That being said, the risk analysis for higher risk class IVDs where the wrong diagnosis would lead to, say, a catastrophic event or the spread of a disease, in that scenario, your risk analysis should be much more stringent versus a device that is not associated with that much risk. You need not have that level of risk analysis as warranted for a medical device. Also, your risk analysis is mainly centered around what happens if your diagnosis goes wrong, what happens if your device does not perform as intended and gives you wrong data, what is the result of having this wrong data. Versus for a medical device, you will be directly talking about what effect or what patient harm a patient experiences if the device malfunctions or it is used incorrectly or it doesn't perform as intended. But similar to medical devices, you do have to perform the risk analysis at the very beginning, at the design stage, at the manufacture stage. The risk analysis should include everything pertaining to shipping, everything pertaining to misuse. So all of those GSPRs will still be applicable here, and all of that has to be covered in the risk analysis and risk management process, but it's not as stringent as the requirements for a medical device. But talking about inadequate design characteristics or immature technology, and then talking about usability studies and defects in manufacturing, all of that is still a requirement under the risk analysis and management for IVDRs.
IVDR mentions, example Annex 9, Chapter 2, the possibility to claim equivalence to another device to use clinical evidence data from this device. What do you think about this approach? Is it a viable way to demonstrate clinical performance? What would be the requirements to claim evidence, equivalence?
So we Stephen, what we've seen is that there have been instances where we haven't even claimed equivalence. We've not claimed that we are equivalent to any device. What we've done is we've done a blanket systematic search looking for the generic name of the analyte that our device identifies or screens or monitors for a specific disease. And then all of the data that has been obtained from this systematic search that supports our device, in the sense that it's the same nature. It's either our device is automated, and what we found in literature also is automated. It works on the same testing population. It works on the same specimen type. In that scenario, all we've done is put up a table showing that the base analyte, the basic materials that are used in developing this in vitro diagnostic are the same, and multiple data sets from multiple different manufacturers, multiple different brands have been leveraged as data for clinical performance, and this has been accepted by the notified body. They've not raised any questions about it. It has to be devices where you can, to a certain extent, prove that they are based on the same characteristic. Like if your device is screening via, say, mass spectrometry, but you are getting data for fluoroscopy, then you can't claim that the evidence is extended to this as well. It has to be the same procedure, the same basic technology, the same. If your IVD has like a clone, it has the same clone as the manufacturer device that you found in your systematic search. If you can establish all of that, then you can definitely claim equivalence, and you can definitely leverage data from the equivalent device or similar devices that you found on the market. Also, the amount of evidence that is required for claiming equivalence is not as stringent as an MDR. You still have to obviously show the clinical, technical and biological similarity, but the number of points that you use to show this similarity is lower in an IVD. As long as you're using the same specimen type, same method of screening or monitoring, you can leverage data from other devices.
Regarding clinical evidence and clinical studies for a Class C genetic test, assuming that there are no recognized geographic differences in the disease status, is it appropriate to include US-based practice guidelines and CAP proficiency data in combination with published literature in place of performing a specific clinical performance study?
If the device is low risk, has been on the market for a considerable amount of time, and you already have sufficient data from published literature, or you already have sufficient data from routine diagnostic testing, you have experience on which you can bank your performance evaluation, there is no requirement to conduct a clinical performance study. It's not a mandatory requirement. But if they feel that the device has not been on the market long enough, if there isn't sufficient published literature, then they will ask for a performance study, but it's not relegated to just European population. US-based studies also are accepted, as long as you can prove that there are no recognized geographical differences.
What is the best approach when a device claims multiple analytes, such as the case of a NGS-based genetic test?
Unfortunately, for devices based on multiple analytes, we have to have proof of scientific validity and proof of clinical performance for each individual analyte. There is no workaround to this. We can't extrapolate data from one analyte to another. However, one thing you can do is it's not always necessary that your combination of analytes be the same as the combination of analytes in other devices in the market. So we have used the approach where devices that have different combinations, as long as they have two or more of the analytes of interest that we are also having in our device, we've leveraged data from those devices as well, and it has been accepted. So we've taken all of the data available either for single analyte or groups of analytes that have two or more of the analytes which our device has, and we've leveraged all of that data as clinical performance data as well as scientific validity data. They've not raised questions on that. It doesn't have to be the same combination as is in your device, and it doesn't have to be always in a combination. You can even leverage data from single analyte basis as well, but each analyte must have enough evidence to establish scientific validity and clinical performance on its own.
All right, we have a couple more questions. Hopefully we can get to them all in the time we have left. Is it a requirement that performance evaluation studies for the IVDR shall be performed within the EU, Europe, or can the studies be performed in the US?
They can be performed in the US as long as you can justify that there are no geographic differences. There's no factors that are influencing the outcome in the US versus the EU.
So for clinical evidence, I can use published literature even if I used it for proof of concept and changed the use of the data. Furthermore, my clinical performance study has to be conducted under ISO 20189.
Sorry, Kelly, can you repeat that?
Oh, sure. The question is, for the clinical evidence, can I use published literature even if I used it for proof of concept and changed the use of the data?
You can use you can use proof of concept studies for your clinical performance data as well, as long as it has that component to it. If your proof of concept studies are demonstrating the clinical utility of the device and not just checking the relation between the analyte and the disease, you can leverage proof of concept studies for your clinical performance as well. But usually proof of concept are they're smaller in, I mean, the data set is going to be small. Since the data set is small, it may not be accepted. So they may ask you to do an additional study. It's not because you're leveraging a proof of concept, it's because they would consider it insufficient amount of data to demonstrate the clinical utility of the device.
All right. They had a second part of their question. The clinical performance study, does it have to be conducted under ISO 20189?
I think the ISO standard was 20916, and yes, if the study is not in accordance with this ISO standard, it won't be accepted.
Okay, thank you. We are at time for our webinar. There are a couple more questions, but I will send them over so that they can be answered virtually, and we have your contact information so that we can get those answered. I want to thank you for attending today and for all the wealth of information from you and Celegence. And thank you so much for joining us today.